Okay. I forgot to record it, of course. God. We're moving on to Chapter 7. This will not be on the exam next week. It's about genetics and inheritance. And these are the objectives. You can read them on your own. We're going to go through all of these things. Okay. In terms of organization, we already talked that the cell, for cellular life, is a fundamental unit. But the cell can be further broken down, obviously. If you're bacteria or archaea, I asked the class whether bacteria and archaea, what's the difference between them and eukaryotic cells? And we also are part of eukarya. And obviously, we, eukarya, have nuclei, have one nucleus, and all eukarya do. And they also have organelles, like mitochondria, ribosomes, and other things that we learned previously. And the nucleus, of course, contains the genetic material, including an identical complement of chromosomes in 2 copies. That means they're homologous chromosomes. And we learned about in cell division how during mitosis, first of all, the DNA is copied, and then so you have twice as much DNA. And then during mitosis or meiosis, you end up splitting and getting 2 cells for mitosis with identical DNA. Whereas with meiosis, you have a lot of variability introduced by sex. Okay. Here's a pair of chromosomes. Humans have 23 pairs of chromosomes. 22 are identical. The other 2 are sex chromosomes. Females are XX, males are XY. As you can see, these homologous chromosomes have genes on them, and genes code for proteins. Here's a gene, and here's a gene. Okay, they're the same genes on the same place on the chromosomes. It could be, though, that they're different versions of a gene or different alleles. So you could also have a situation here where you have that. And here, a different allele. In each chromosome is one long DNA molecule, unlike bacteria and archaea, which have circular DNA. Eukaryotes have long strand of DNA. And they actually have 2. One from mom, one from pop. And the genes are the functional regions in that they code for proteins. And the DNA is a double helix, and in between each side are 2 base pairs, A and T go together, and C and G. You always find that. You never find an A and a C or a T and a G. Okay. Okay, so I mentioned we had 23 homologous pairs of chromosomes. Homologous means same. Locus means genes at specific spots on the chromosome. Those genes are in the same place on each chromosome. You inherit one chromosome from mom, one chromosome from dad. Each gene, you can inherit one of two one allele. They could be the same allele, like mom and dad. Could both have, let's say, a gene for black hair, and the offspring will have black hair. It'll be for a pigment, a protein pigment called melanin. Or it could be that, let's say, dad actually has blonde hair, so the offspring would have one blonde hair pigment, one blonde hair allele, and then one black hair allele. Okay? We've gone over homologous pairs before, chromosomes, one from mom, one from two. We have 23 homologous chromosomes, pairs of homologous chromosomes, so we have a total of 46. During meiosis, we end up, each sperm or egg has 23 single chromosomes. If it was from the dad, in the sperm, 23 chromosomes from dad. The egg, 23 chromosomes, single chromosomes from mom. The discovery of the structure of DNA, you can watch this YouTube if you want, right there. It's a pretty interesting story. It's about 15 minutes long. People had been searching for the molecular basis for inheritance for a long time. It wasn't. They finally mostly figured out that it was probably DNA, because by this time they could see during mitosis and meiosis, the chromosomes condensing and then going through each stage of mitosis, including then along the metaphase, and then pulling apart, and then finally two different identical cells. So they figured that was actually probably the molecule of that, but they didn't know what the structure was. And so in 1953, Francis Crick and James Watson, who were at Oxford, they had an advisor who, told them what to do and what they should focus on, and they ignored him. And they wanted to do something important, and so they decided to try to discover the structure of DNA. And there were other labs focusing on this too, but they were the first to do it and received the Nobel Prize for it. And then later, Francis Crick worked out the genetic code of the DNA in terms of how DNA was transcribed and translated into proteins or amino acids, which make up proteins. Okay. Our chromosomes are made up of DNA, and they have a backbone of phosphate and sugars. And then in between, they have these DNA bases. Okay, and let's see. Okay. The bases are A, T, C, and G. Okay. As you can see, adenine always goes with thymine, and cytosine always goes with guanine. Look to the right. We see this. So here's the double helix. These are the sugars and phosphates, right? And those are the phosphates, and here's the sugars. And then the bases. A and T, adenine and thymine, C and G, or it could be G and C and T and A. Doesn't matter. They biochemically fit together. And those bases are what is actually read in terms of turning that information into amino acids and proteins. They always come in pairs. So let's say we have an A and we have a T here. Or we might have a C and a G there. Okay? So if you have, let's say, an A here, you know automatically that its complement is thymine here. Oh, fuck. Come on! Okay, there we go. Okay. During copying, the double-stranded DNA molecule unwinds. There's an enzyme, DNA polymerase, that adds. So if you have one side, you can always make the complement, because you know always that A goes with T, C goes with G. And that's how you end up with two new molecules during mitosis. And we have a mutation. This mutation goes from A to T. And this mutation goes from T to A. Or this goes C to G. Or G to C. Okay? So those are mutations, and that can affect which proteins are made, and that can affect on the organism in terms of it might be bad for the organism, it might give you a protein that doesn't work, it might be good for an organism, beneficial, or because there are so many bases. There's 3 billion base pairs, but only 20,000 genes. Some of those genes have millions of base pairs long, but there's a lot of DNA that doesn't code for proteins per se. So some mutations have no effect. Okay. A genetic trait is any inherited characteristic of an organism that can be observed or detected. The genes, though, don't code for traits specifically. They code for proteins with certain characteristics. And those characteristics are what we see as traits. So we're going to learn about genotype, the genetics underlying the proteins, and phenotype, the proteins underlying the traits. There are some traits that are invariant. That means they're unchanging. For example, all humans are born with 4 limbs. Unless there's a mutation, but it's an invariant trait. Okay? We have 2 arms and 2 legs. We have 5 fingers on one hand, 5 fingers on the other. We have certain bones associated with each. Those are invariant traits. There are other vertebrates that live on the land that also have 4 limbs, 4 appendages. They are called tetrapods, and they include salamanders, lizards, tortoises and turtles, crocodiles, birds, and dinosaurs, which are extinct except for birds. They all have 4 limbs. The limbs are made of certain bones. The same bones are used in each of these different groups of organisms. Why would that be? Well, it turns out they inherited this body plan that involves 4 limbs. So, in other words, the genetics are responsible for tetrapod body plans. In other words, 4 limbs. Okay? Now, there's some that have lost 4 limbs, like snakes, but we know that they're still vertebrates and that their ancestors had 4 limbs because we can look at their skeleton and we can actually see in some cases little legs and shoulders and pelvises and all that. And the same thing we can see with cetaceans, whales, and porpoises. They too came from an animal that had 4 limbs on the land, and then their ancestors evolved and lost those limbs and went and ended up living in the sea. Okay? Those are invariant traits. Variant traits are traits that differ among individuals. Let's say the color of your eyes, the color of your hair, how tall you are, how smart you are. There's variation in all these traits, okay, that differ among individuals in the same species. So we're all humans, but we have all different kinds of variant traits. Okay. And we're going to learn about the basis for those traits. Now, physical traits, they can either be invariant or variant. Are easily observable traits: hair color, how tall you are, how fast you can run, all these kinds of things. There are biochemical traits, like your metabolic rate, how you process glucose, the enzymes underlying how well you exercise, all that. And then there's also behavioral traits. In other words, how you act, and also, it turns out, how you think and all that. That there's a genetic basis for those in terms of the proteins that are underlying them. It says here, behavioral traits, behaviors with some genetic basis. Well, I would say no. Behavior is just like biochemical, just like physical. The only reason that they're saying behavioral basis is genetic, well, first of all traits generally have a genetic and an environmental basis in that the genes interact with the environment and produce a protein with certain traits. Okay? And so it used to be thought that mind and matter were separate. We now know that the basis for mind, for how we think and act, is material. Okay? So there's a genetic basis for behavior and intellect and all that. Okay. Okay. The gene is a basic unit of information affecting a trait that has a genetic basis. In us, it consists of a stretch of DNA coding for a protein on a chromosome. However, there is not a one-to-one correspondence between a gene and one protein. Initially, about 10 years ago, there was a human genome project to discover and map out on all our chromosomes all the different genes. And there were, by that time, over a couple of 100,000 proteins had been discovered. And people thought that there was one gene for every protein. And when they finished sequencing the entire genome, they only found 20,000 genes. This upended our understanding of the genetic code, because obviously there should only be 20,000 proteins, but there are hundreds of thousands of different proteins in the human body. It turns out genes often code for a bunch of related different proteins depending on the structure of the gene. And that is responsible for which proteins are made and which variants of proteins are made. And there are large stretches of the DNA that in our genome that don't code for proteins, but it turns out they regulate which proteins are coded for depending on which genes are expressed or turned on and off. Okay? Now, I mentioned, and I asked this in class, what is an allele? And an allele is a different version of a gene. So, let's say we have a gene for the color of flowers. We'll say red. Okay? And we also have an allele for the color blue. We have two alleles. In some cases, there may be thousands of different versions of a gene, but since we're diploid, we get one allele from mom and one allele from dad. If both mom and dad have red allele and we're diploid, we will be RR, or if we're both BB, or we could end up with RB because we're diploid. Now, there might be red, blue, another allele that we don't see here, green, yellow, right? Two other people, the dad may have a yellow, and the mom yellow, green, yellow, green, or, oh shoot, green. And they may produce offspring that are green and yellow. Okay. Finally, a mutation is any change in the DNA sequence that makes up a gene, in other words, the bases. So, let's say we go back. Oh. Okay. So here we are. Each homologous pair of chromosomes consists of one chromosome inherited from each parent. The genes are segments of DNA that encode the information for not a trait, but a protein. Actually, they code for so the genes of DNA, they code for amino acids which equals a protein. Okay. Now, okay. So there are two alleles. As I mentioned earlier, You could have the same alleles, one from mom, one from dad. Let's say gene A here. And we can say it's blue. And so we say both mom and dad have the same allele there, blue. Or the genes are in the same place along the chromosomes, but let's say dad has a yellow allele and mom has a green allele, right? Okay. Oh, and in eukaryotes, of course, chromosomes or a DNA is long strands of chromosomes, not like prokaryotes would have circular. Okay? And then the molecule is a double helix with the base pairs inside. And this is what is copied either during mitosis, when cells are replicating, or meiosis, when gametes are forming. And gametes are haploid. They only have one chromosome, not two, like a diploid organism like our cells. So a haploid gamete needs another haploid gamete, in this case a sperm and an egg, fertilizing the egg. And then you have diploid again. Okay. So what's the physical structure of the gene? Well, it's made of DNA, and the coding is according to base pairs, nitrogen-based base pairs, that then code for amino acids and eventually proteins. How many copies of each gene are found in the diploid cells in a woman's body? Well, a woman is a diploid organism. She makes haploid eggs, but since she's diploid, she will have two copies of each gene. They may be the same copies, the same alleles, or they may be different. She has one from her mom and one from her dad. With 46 chromosomes in a human diploid cell, oh, by the way, of course, a man is also completely diploid, and he also has a full complement of 23 homologous pairs of chromosomes, or 46 chromosomes. Not all organisms actually have a dominant diploid life cycle. Some mix it up. So like bees, some bees and wasps and ants, the females are XX. They have two sex chromosomes, X and X. But the male, in that case, instead of being XY, they only have one chromosome from their mother, and they're haploid. But that's just so you know, it doesn't always have to be diploid. But for humans and most other animals, it's diploid, adults, and haploid gametes. So with 40 chromosomes in a human diploid cell, how many chromosomes come from the person's mother, and how many chromosomes from the father? Well, each mother has 46 chromosomes. In terms of offspring, her haploid eggs will have 23 single chromosomes, and the sperm will have 23 single chromosomes. Those 23, when they come together, have the same genes on each chromosome, but they may have different alleles. Okay. Phenotype versus genotype. First of all, let's just look at genotype. Genotype is the genes underneath. So let's say you have this is a strand of DNA. A T G C G G. Okay? It turns out that 3 base pairs code for 1 amino acid. Okay? So we could have AAG, and the complement could be A, well, actually, let's say, no, sorry. This is from mom and dad. Okay. So, obviously the complement for this A is a T. Okay. The mom could have an A in this base pair, and her other chromosome could have a T. Or she gets an A, this individual gets an A from her mom and a T from her dad. Okay. 3 base pairs code for a specific protein. They code for actually a specific amino acid, and amino acids are then strung together in long chains to form a protein. Okay, so this is a genotype, AAG, G, C, T. Okay, now, the phenotype is what you can observe, in other words, the trait, but the trait, these don't code for traits. AAG codes for an amino acid, and then T codes for another amino acid, right? And then if you string these amino acids together, you end up with a protein. The protein gives rise to the traits, the physical expression of a gene. And that physical expression can be physical, like let's say eye color. So you're coding for protein pigments that have specific colors to them. Or it could be a physiological trait. In other words, do they produce lactase and therefore are lactose tolerant or not? That's physiological, whether they can break down milk products or not. Or behavior, the same thing, could be a behavioral trait. Okay? So traits are what we can either see or hear or measure, whatever, physiology, all that kind of stuff. But the underlying basis for those traits is the proteins that are made and instructed to be made from the genetic code. So, poodles illustrate variation in the coat color gene. We have 2 poodles here, one with a brown coat, one with a black coat. The phenotype is the coat color. Okay? The basis for this phenotype, it could be other things. Let's say we decided that the person decided to dye the hair of these poodles. Then the basis would be because of the dye. But in this case, the color is due to the protein pigments that are responsible for coat color, and those are coded for in the genotype. And so, in this case, phenotype, we have brown or black. And we'll say that black is large B and brown is small b. And then the genotypes, in this case, is a dominant expression pattern. Okay, so here's the genotype, the underlying genes, and for brown, it's recessive, so you need 2 small b's. But for black, you either need 1 large B, 2 large B's, or 1 large B and 1 small b. Okay? Sometimes you could end up with a dark brown. That's a different expression pattern called incomplete dominance, what we'll get to. But for now, we're just going to talk about dominant and recessive traits. Okay? And underlying coat color are protein pigments, and the basis for the protein pigments are due to the genotype in terms of the arrangements of base pairs. In this case, large B is dominant. Okay. So, the dominant allele is an allele, a variant of a gene, that is expressed when there are, let's say, 2 copies of it. Well, that's not really true. That's misleading. Okay? A dominant trait, as long as there is 1 large B, will be the dominant trait. So, a dominant could be B, B, or B, little b, but it will be a black coat color. Okay? Now, so both of these individuals, this and this, will give rise to this black dog. Okay? Now, the only way you're going to get a brown dog is if you have B and B. Then you're going to have a brown dog. Okay. If you have B, B, what color will it be? It will always be black. Okay? It's only when you have BB, little b, that you will have brown. So little b is a recessive allele. Big B is the dominant allele. Dominant, whether it's 2 large B's or B large B, little b will still express the big B phenotype, which is black. We're talking about dogs. Now, we've got some big words that go with this. Okay, so homozygous. Homo means the same. Zygosity has to do with the zygote, meaning having zygotes of a specific kind. So if you have the same zygotes that are diploid, they have the same alleles. Okay? So homozygous equals same okay? And in this case, if large B is dominant and the individual is homozygous, they are B. Okay. You could also, of course, have an individual that's homozygous recessive. If this was a dog and large B was the dominant for black coat and small b was the recessive for brown coat, and you had two small b's, you would have a brown dog. So here you would have a black dog, and here you would have a brown dog. Okay, they are both homozygous, but with different alleles. Since B, large B, is dominant, if you have an individual with a large B and a small b, remember, black is dominant, and so you would end up also with a black dog. It's only with small b's that you would end up with a brown dog. Okay? Heterozygous, if it's dominant expression pattern, the phenotype expresses the dominant allele, in this case, a protein with a black pigment. A dominant phenotype, all you need is one dominant allele, in this case, in terms of black and brown dogs, right? Either two black alleles or a black and a brown allele, and ends up a black dog, or the only way you get the recessive trait, in this case a brown dog, is two brown alleles. Individuals that are heterozygous, black, big B, little b, end up with a black dog, in this case because it's a dominant, big B is a dominant. Sorry. Which might you observe directly, the genotype or the phenotype? Can you see what genes these dogs have? No. You can only observe the phenotype. You don't even know what the basis is for their color. it could be that someone has dyed these dogs, and originally, let's say they had white fur, and then, so you can't even tell whether the basis for the color in terms of structural proteins or a dye or something, right? Which poodle could be heterozygous? The one with a black coat or the brown coat? Is it this individual who could be heterozygous, or is it this individual who could be heterozygous? Remember, black is dominant. If you have one copy, you will be black. Here's our black dog. Okay. And that individual could either be B or B. B. Okay? So which poodle could be heterozygous in this case? This one only. Okay? Can you identify which, with certainty, the genotype of a black poodle? The genotype are the alleles, either large B or small b. Can you, absolutely with certainty, identify whether this is homozygous dominant or heterozygous, large B, small b? No, you can't with a black poodle. With a brown poodle, you can. The only way you can get a brown poodle is by two small b's okay? You cannot get a brown poodle from a heterozygote. Only homozygote recessive. If we remember, okay, Gregor Mendel was a Czech monk. He lived in the what is now the Czech Republic. It was actually part of the Holy Roman Empire back then, and the Habsburg Empire. But anyway, he was, I don't know, Franciscan or Benedictine monk. Part of Catholicism is doing good deeds, and he was an agriculturalist, and he wanted to find out about how to make sure that plants bred true. So, for example, let's say you wanted good corn with nice large kernels. How would you make sure that you had seed that would actually result in corn with nice large kernels versus some varieties that have very small kernels? Okay, so he wanted to understand this. Now, at this time, this was 1860s, 1850s, 1860s. This was the time when Darwin was coming up with publishing his theory of evolution by natural selection. And at this time, most people believed in what's called blending inheritance. Mendel, on the other hand, in this case snapdragons, but he also worked with pea plants. He started crossing parents of known color flowers. Okay? And in snapdragons, in fact, you end up with incomplete dominance. In other words, pink flowers. Well, okay. Pink flower. But he also worked with pea plants, and there was dominance and recessiveness like we were talking about earlier. And he did crosses. And with pea plants, he ended up with either all red flowers or all white flowers, but in an unequal ratio. Okay? So, he had an experimental garden. He performed genetic crosses, which are controlled mating or manipulative experiments that can determine how traits are inherited. Okay. And because of this problem with blending inheritance, Darwin had a problem. So he didn't know how inheritance actually happened. He had a misinformed understanding of inheritance. No one knew until it was Gregor Mendel that came along. And even then, even though he actually had Darwin's book on the origin of species, he didn't see how it applied to that book. And unfortunately, he only published in an agricultural journal, and then his material was lost. No one read it, and it wasn't until 50 years later, in the early 1900s, that people figured the same thing out and then rediscovered that he had actually discovered it first in the 1860s. Okay? Okay? Okay. Hold on. Okay, so here we have blending inheritance. We have a vase, two vases. Okay? Okay. Now, it was thought in terms of traits, in this case, proteins that give organisms certain characteristics. In this case, we'll say coat color, or we'll say plants. And let's say we have two kinds of paint. We've got blue paint, and we pour some in here, and we have yellow paint, and we pour some in here, and they mix together, and we end up with green paint. Now let's say we were in an environment where blue was beneficial. If you had individual with parents who were, let's say, blue, or yellow, and you formed an offspring that was blue, yellow, you would end up with a green offspring, and in this case, they would be suboptimal. In other words, it would dilute the fitness of the individual, right? Okay, now, Okay. Genetic crosses. This is what Mendel did with his flowers. In this case, pea plants. And pea plants are dominant recessive. Okay. The same thing is going on in terms of genetic crosses that Mendel was doing. Okay. So, in this case, instead of big P, little p, we're going to have two different flower colors, red and white. Okay? So, we're going to say red is dominant, and if it's homozygous, it's RR. And if it's homozygous recessive, white, it has to be WW. Okay? Now, if we were to cross this, remember, the father, let's say the father is red, so, and we know he breeds true. He always produces a red offspring. And the mother always produces a white offspring. So, the father donor is always going to donate a red to the offspring, because that's all he has. He donates this red, or this red, it doesn't matter. Eventually, you're going to end up with an offspring that has a red from dad. The same thing is happening for mom. She can donate this white allele or this white allele, and so the first F1 generation is going to be heterozygous. That means red-white alleles, not red-red or white-white. And the red comes from dad, the white comes from mom. If we were to then cross heterozygous flower color, in other words, an R and a W, we would end up as follows. Okay, this is called Punnett square. And for simple genetic crosses, this is how we figure out percentages. Okay? Okay. So, on the left, first of all, is dad in parental generation. Okay, so dad is red, R, R, and that's father. Okay, mother is white, and W, even though I'm using black. Okay, so the parental, we were always going to get R, 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 and W, 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 right? This is equal to the F1. Okay. Here's our Punnett square again. Here's father, Here's mother. Okay. Remember now, everybody's heterozygous. So father is R, W, and mother is RW. And their offspring, because R, red is dominant, are all red flowers. Okay? So let's cross them. Each one of these is a separate allele, right? They can either donate, the father can donate either an R or a W, and the same thing with mom. Okay, so we have RR. Father donates an R, mother donates an R. In this case, for this square on the right, we have R W, because mom donates a W. And here, father donates a W, and mother donates. Father donates a W, and mother donates an R. Or finally, here we have W, W. Okay. So, R is dominant. So, RR equals red flower, or RW equals red flower. It's only WW that equals white flower, right? So whether it's the first one is homozygous dominant RR. What kind of flower is that? It's a red flower. Okay. Then RW, that's the R from dad and the W from mom. What color flower will that be? It's also red. Okay. Here we have WR. Doesn't really matter. In this case, the dad gave the W and the mom gave the red instead of the dad giving the R and the mom giving the W. Okay? And so, again, we end up with a red. It is only when both ended up giving the recessive W. So, hold on a second. Okay. That we ended up with a white flower. And there we have it. Okay? So, this is a 3 to... hold on. This is a 3 to 1 ratio if red is dominant. Okay, so this is the F2 generation. Okay, now let's go up. So we now have first of all, P, small p, the crosses result in large P, small p. And then these crosses, P, right, end up, when they planted the seeds that grew into F1 plants, he allowed those plants to then self-fertilize and produce the F2 generation. So, Dad, in this case, or Mom, could have a large P or a small p. The same thing with the other donor, Mom, large P, small p. And then, if we did the cross, we would end up, again, with P, P, oops, undo, little p, big P, or P. That's the F2. This, of course, is a 3, 2, 1. So you always find this pattern when you have both parents heterozygous dominant. Okay? The dominant allele is going to show 75% of the time, the recessive allele 25% of the time. Okay, so what would you predict about the color of the F1 plants? Remember, we have parents who were P and little p, and so the F1 plants would be big P, little p. Okay. Why was it important that Mendel begin with pea plants that he knew bred true for flower color? Well, first of all, you can't be certain in terms of dominance, but you can be certain in terms of recessive. So he knew he had recessives for sure. And then when he bred some plants with a what he knew were two heterozygous, he would always get a dominant. Okay? And so he knew one of the plants was homozygous dominant. Okay? So he had to make sure that it was always, anytime it bred, it was always going to produce a dominant offspring, in this case, purple flowers. If he didn't know that, then he wouldn't be able to predict what was going to actually happen. Okay? So, he knew he had purebred purple flowers and purebred white flowers. Why couldn't he simply cross a purple flowered plant and a white-flowered plant? Well, the white-flowered plant is fine, but the purple, you can't tell by looking at them whether it's large P, or large P, small p, heterozygous. Okay. So again, I'll take this, we'll do it again. No, these are, okay, we have a Punnett square. We're using large P and small p now. Okay. Over here is the father. Here's the mother. Okay. We know that they're both heterozygous. Parental generation. So, father is large P. Mother is small p. Okay, and here we then cross them, and we have P. This is the F1 generation. Okay, so here's the F1 generation, and we're going to interbreed them. We know the F1 generation is all heterozygous dominant, so they're going to have purple flowers. So all the individuals, first of all, let's do a Punnett square. Father, mother. Father is P, mother is P. So for the F2, P, purple, P, what color? Purple, P, what color? Purple, what color? Small p, white. Okay, so for this, we have purple flower. For this, we have purple flower. For this, we have purple flower. And for this, we have a white flower. Okay, so again, the offspring, the F1 offspring of parentals, large P, small p, are always heterozygous, large P, small p. Okay? Each egg and each sperm produced by the F1 plants has a 50% chance of receiving a P allele, large P, and a 50% chance of receiving a small p allele. Okay. Egg and sperm can combine in 4 possible ways in the F2 generation. Now remember, in meiosis, you're producing haploid gametes that are either sperm or egg, or in this case, egg or pollen, ovary or pollen. Okay? And if you remember, in meiosis, you can have 4 possible outcomes. Okay, a lot more than that, but if you only had 2 alleles. Okay. The Punnett square method predicts 3 purple flowered offspring for every 1 white offspring. So, for example, large P, we can either have the F2. Why did Mendel's entire F1 generation look the same? Because they were P. Okay. The phenotype ratio in the F2 generation is 3:1. So we either have P, homozygous dominant, P, little b, heterozygous, but still purple, again, heterozygous, still purple, and then finally, white, P. So draw a Punnett square for a genetic cross of 2 heterozygous black coated dogs. Okay, remember, they've got a black and a brown. Okay, so what are the expected phenotype and genotype ratios? Here's our Punnett square for puppies. Okay. And we know they're all heterozygotes, black. Okay, but they're heterozygotes. So let's say male and female. And the male is large B for black. And small b for brown. And the same thing for the female. Okay. So we have our crosses. We have large B, and we have a large B here, a large B here, and then we have browns. Okay. And then we have black. We have black. Here we have black. Okay. Okay. So we have homozygous dominant. This. We have heterozygous dominant. Both of these. And we get homozygous recessive, these.